Hey guys, today we are going to be talking about the general body plans and taxonomy of sponges. So this is the first phylum of animals for the school year, and the name Porifera means pore-bearing in Latin. So when we think about the basic characteristics of sponges, they are asymmetric, meaning that they don't have any symmetry, and they also don't have any tissues or organs. These animals are sessile, which means that they don't move as adults, and they get their nutrients through filter feeding. So if you're looking for sponges, they can be found in both freshwater and saltwater environments. The ones you probably know about are the super bright, pretty corals that you find in saltwater. It's less common to know about the freshwater sponges that are usually a dull greenish color. So when we look at sponges, they are the most simple of all animals. There's no nervous system, no organ system. They also don't have any tissue organization. So we're going to talk about through these notes all of the different ways that sponges function. The first one, if you remember from previous pictures, there was always one really large hole or opening at the top of the sponge. This is called the osculum, and that's where water is leaving through the sponge. So water will be brought into their bodies through little pores along the outside, pumped through their system, and leave through that osculum. So when you look at the body structure of sponges, we have three basic types. We have ascon, cycon, and leucon. So ascon is very basic. And this is the most simple type of organization. So water is going to be entering through the pores on that tube-shaped sponge, and then it'll be expelled through one single opening, or the osculum. Then we have cycon. Cycon is similar to ascon, with a single opening, a single osculum for water to diffuse out of. But there are more infoldings and a little bit more complex canals for water to move through before it leaves through that osculum. Then the most complex body type, we have leucon. So not all of the water is going to move through the same canals and leave through the same osculum in leucon. There are a series of canals and chambers that water is going to move through before it's expelled through one of the oscula. This is the largest kind of sponge and the most common body type of sponges. Now we're going to get more detailed and talk about some of the major cell types. So there are four major cell types that we're going to focus on. They are the pinacocytes, the porocytes, amoebocytes, and choanocytes. So pinacocytes, they're basically the epithelial cells. So they're what run along the outside of the sponge. Then we have, so this would be a pinacocyte, and this would be a pinacocyte. Then we have the porocytes. These are your tube-like cells, and they're going to help control the flow of water. So if we look right here, we see a bunch of porocytes. So we've got a porocyte right here, and we see that water is moving through it. Then we have these also help not only with the flow of water, but circulation through the sponge. Then we have amoebocytes. So those are going to differentiate into other cell types. And then the last one you need to know for now, you need to know what it is and where they're found, are the choanocytes. So choanocytes, they're also known as collar cells, and you need to notice that they're flagellated. So that flagellated structure, it's going to help create currents needed for filter feeding, and then that collar portion of the cell right here, it's going to help trap food particles. So this diagram, or these diagrams, they help show us those cells that we just talked about again, and there are a few more details with the cell types. So we already talked about those amoebocytes. We already talked about the pinacocytes, so we can kind of see them better right here. We see the cells that are on the outside of the sponge. And we also already talked about choanocytes, or collar cells. We talked about porocytes as well, but we don't see them labeled. The reason why is if you look for the opening where water goes through, it's right here, that is the ostia, and ostia are made out of these porocyte cells. So there are three different structures that you need to know about how sponges support their bodies. The first one is the mesohele. This is non-cellular, and it's between the inner and outer tissue layers. So the inner material, the mesohele, it's that inner material, and it makes up the main body of a sponge, and it's a gel-like mixture. Now, next one, you're probably familiar with spongin, even if you don't realize it. So spongin is what allows a sponge, such as one you may have found at the beach, let's say it washed up on the beach, it's going to be soft and flexible. So if you've ever touched a sponge that's harder and it feels more pointy, you're feeling the spicules. So spicules, they come in many shapes like we see here. And they're made out of some sort of limestone or silica. We'll talk about that in a little bit of how limestone is calcium carbonate. So since sponges are so simple, you might ask yourself, how do they eat and what do they eat? So they eat things that are in the water, such as bacteria, algae, protists that are entering through their pores. So first what's going to happen is food is going to enter through those porocytes or the ostia, and then they're going to move to the choanocyte, which is this collar cell right here. If you look at the diagram, the flagella, we see arrows. Arrows are showing that the flagella are moving, and they're helping create a water current that actually moves the food particles into a vacuole down into the bottom of the cell. And then that food is going to be broken down and used as nutrition for the sponge. There are two ways that we can get sponges, that they can arise. First one we're going to talk about is asexual reproduction. So there are a few ways that this can happen. When a sponge buds, so budding, a new sponge is going to begin to grow. So we can actually see budding occurring in this picture right here. So a new sponge is going to start to grow, and then once it reaches maturity, it's going to separate from that original sponge. The next one is regeneration, and this one people usually find to be the coolest way. So regeneration can occur if a piece of sponge breaks off. A new sponge is going to grow from that piece that broke off. And then the last thing, which personally I think is the coolest, are gemmules. So gemmules, they are these internal survival pods that are there in case a sponge is in some sort of hostile environment. So the gemmule cells, they're going to survive because they're surrounded by a very protective spicule layer, and that gemmule is just going to stay dormant until the environment is acceptable for sponge growth again. So once the environment is favorable, then that gemmule is going to start to grow into a new sponge. Next, we're going to talk about sexual reproduction. So believe it or not, sponges do release sperm and egg cells. That's actually what's happening right here. So a single sponge, it's going to produce both gamete types, which is called being hermaphroditic. So a key thing to note is a sponge is not going to self-fertilize its own eggs. The main reason why is because the gametes are created at different times and by different cell types, which we can sort of see right here. So choanocytes, they are going to produce sperm, and archaeocytes, they are going to produce the eggs. It's a good thing that sponges cannot self-fertilize because sexual reproduction, the whole point is to increase that genetic diversity. So once a sponge has an egg that is ready to be fertilized, that egg will be fertilized in the mesohele that we can see right here. I definitely recommend pausing and taking a look at this diagram. Here's another great diagram on the left right here showing us the sponge life cycle. So again, I recommend pausing the video and labeling all of these diagrams on your guided notes. An important thing to note is that this larval stage is not sessile. So larvae of sponges are able to, move, they're mobile, until they attach to some sort of surface, like we see in this step right here, to then begin to develop into a mature sponge. Now that we've talked about a ton of cell types and body plans of sponges, we're going to touch on how do we classify sponges. So they are actually classified on their skeleton type. If there are silicon-like spicules, so these right here, then the sponge is going to be in the class Hexactinellida, and that could be something like a glass sponge. If the sponge has both silica-like spicules and spongin fibers, then it's going to be in Demospongy, which contains, this is about 95% of all of the sponges that we find today. And then the last type are your calcareous skeletons. So these are your class Calcarea sponges. We're going to break down our sponge classes a little bit more. So when you look at class Calcarea, these are very small marine sponges, so you are not going to find any Calcarea sponges in freshwater. Then class Demospongi, like we said on the last slide, this is what most sponges are, and they have the most complex body type, which if you remember is leucon. And then the last class that we'll talk about is the Hexactinolida. And these are your glass sponges. They're really pretty. They're made out of spicules, and they come in a cup, case, or urn shape. Now, last thing we're going to talk about, you are almost done, with sponges is a little bit of sponge ecology. So if you were paying attention, then you probably noticed that all of the other sponges in this video were attached to other objects. This is because, like we've said before, they are sessile, and attachment can be anything on rocks, corals, or any other just submerged object, honestly. So you may have seen recent reef rebuilding projects where they place some strange looking objects on the bottom of the ocean. This is to serve as a base for those coral reefs and for things like sponges to attach to and then start growing again. All right, so I recommend pausing the video right here, and after everything that we've talked about for sponges, you should be able to answer these questions. So find out, how much did you sponge up? And I will see you next time.